Good morning, students. Today we will continue the topic acid-base balance computation number 6.7, and today's topic is regulation of blood pH. So the pH of blood is regulated by three systems. First is buffers, second respiratory system, and last is kidney. So we will study one by one each of these. First, you should know the normal pH of blood or plasma is 7.4. It ranges from 7.35 to 7.45. When the pH falls below 7.35, it is called as acidosis, and when it increases above 7.45, it is called as alkalosis. Both have some deleterious effect on our body. Like acidosis leads to CNS depression and coma, whereas alkalosis leads to hyperexcitability and tetany. In body, acids are formed, so pH is regulated by neutralizing these acids. That means there is constant production of acids takes place in our body. So pH of the body is regulated by three different ways, as I have told you: buffer system, respiratory system, and kidney, that is renal mechanism. So first we will study the role of blood buffers. It is also called as first line of defense against the change in pH. These buffers neutralize the acids by the base component present in the buffer system. When the base component is depleted, the buffer becomes ineffective in neutralizing the acids, and then they should be replenished. The important buffer systems of our body are bicarbonate buffer system, phosphate buffer system, and protein buffer system. First is bicarbonate buffer system. It is the main buffer system of extracellular fluid, means outside the cell. The composition of this buffer system is bicarbonate and carbonic acid. The ratio is 20:1. That means bicarbonate is 20 times more than carbonic acid. In this buffer system, if you see, base is 20 times more than acid, therefore it is more effective to neutralize the acids, various acids forms in the body. This bicarbonate is also called as alkali reserve of the body, which is sufficiently high to meet the acid loads of the body. If you go to the actual concentration of carbonic acid is 1.2 millimole per liter in normal circumstances, and the concentration of bicarbonate is 24 millimole per liter in normal situation. In this buffer system, carbonic acid is regulated by respiratory system and bicarbonate is regulated by kidney. Hence, this buffer system is very effective. It accounts for 65% of the buffering capacity in plasma and 40% buffering action in the whole body. How it acts? Very simple. When the acids are added to the blood or in the extracellular fluid, it is shown here by H+ ion. The bicarbonate or base of this bicarbonate buffer system combines with it and converted into H2CO3. On the contrary, if bases, which is represented by A- here, are added to the extracellular fluid, then the acid part of this bicarbonate buffer system, that is H2CO3, combines with it and neutralizes. So in this way, this buffer system acts when acids or bases are added to the extracellular fluid. After completing the bicarbonate buffer system, we will study phosphate buffer system. It is the main buffer system of intracellular fluid. Its concentration in plasma is low. The composition of this buffer system is Na2HPO4, which represented the base, whereas NaH2PO4, which represent the acid portion of the buffer system. The ratio is 4:1. So here again the base is 4 times more than acid, and just now I have told you why base is more, we require more base, because there are constant production of acids occurs in the body in the form of ketone bodies or intermediates of various cycles. So it is also very effective buffer system because its pK value is 6.8, which is nearer to physiological pH. And what is pK? As we have studied in the previous lecture, pK is nothing but it is a pH at which the buffer system is more effective. So pK is of two types, pKa and pKb. pKa means the pH in acidic range, and pKb means it is the pH in basic range, where the buffer system is more effective. Protein buffer system. All of us know proteins are made up of amino acids. Each amino acids have side chains in the form of R. We represent it by R. Buffering capacity of protein depends on pKa value of ionizable side chains. The most effective group is histidine, in imidazole group of histidine, which has pK value 6.1. Hence, protein with more number of histidine residue is more effective buffer at physiological pH 7.4. Proteins in the intracellular fluid, hemoglobin in the red blood cells, and albumin in the plasma act as a buffer due to presence of amino acid histidine. What is the specialty of histidine? Histidine has a imidazole group as a side chain, and that imidazole group has pK value 6.1, which is nearer to physiological pH. The examples of protein buffer system is given here. Protein minus is the base, H protein is the acid, HB minus is the base, HHB is the base acid, albumin minus is the base, and H albumin is the acid. So hemoglobin has 38 histidine residues and albumin has 16 histidine amino acids residues. Hence hemoglobin has more histidine, so hemoglobin is more effective buffer system as compared to albumin. Now it is said that buffer acts quickly but not permanently. Why? Because buffer can respond immediately on addition of acid or base, but they do not serve to eliminate this acid from the body. That means they neutralize the acids or base, but they will not eliminate that acid from the body. For example, if H plus are added, the bicarbonate base of the bicarbonate buffer system combines with it and neutralize that H plus ion. But in that process, the bicarbonate is utilized. So that alkali reserve will not be replenished by the buffer system. So for final elimination of these acids, the respiratory system and renal regulations are very, very essential. Now we will study the role of respiratory system in regulation of blood pH. It is called as second line of defense against change in pH. It regulates the body pH by regulating the concentration of volatile acid like H2CO3, that is carbonic acid. When the concentration of H plus ions increases in the body, it combines with the bicarbonate ions to form H2CO3 in the presence of enzyme carbonic anhydrase to form carbonic acid. So when the concentration of H2CO3 increases, it dissociates into carbon dioxide and water, again in the presence of same enzyme carbonic anhydrase. So overall reaction is given below. Here H plus ions combines with bicarbonate ion to form H2CO3, and that H2CO3 is broken down into CO2 and water. Now the concentration from this reaction, you can state that the concentration of carbon dioxide, carbonic acid and H plus ion are in equilibrium. These reactions are reversible reaction. So the concentration of these three compounds, carbon dioxide, carbonic acid and H plus ion, are in equilibrium. Remember this. That means when the concentration of one increases, other will also increases, and vice versa. So here it is given, when the concentration of H plus ion increases, pH falls and carbon dioxide also increases because of that reaction. So what will happen if carbon dioxide increases? It stimulates the respiratory system. The rate and depth of respiration increases and carbon dioxide is removed from the body. So in this way, the respiratory system plays an important role in regulation of blood pH. On the contrary, when the concentration of H plus ion decreases, pH decreases and the concentration of carbon dioxide decreases, because H plus ion and carbon dioxide are in equilibrium by this reaction. So when the concentration of carbon dioxide decreases, it decreases the respiratory, or it inhibits the respiratory center, and the rate of respiration decreases and carbon dioxide is retained in the body. So this is about the regulation of pH by respiratory system. And last and important is role of kidney in regulation of blood pH. This is very important. The main target of kidney is to regulate the pH of extracellular fluid. So it is also related to bicarbonate buffer system of the extracellular fluid. The main aim of kidney is excretion of H plus ion in the urine without change in pH of the urine, means without much change in the pH of urine. What happens if pH of urine changes or decreases too much? It may damage the urinary tract. So the first aim is very clear: H plus ion should be excreted, but pH should not be decreased to a large extent. And second aim is regeneration and reabsorption of bicarbonate ions, which is also called as alkali reserve of the body. Due to excretion of acids in the urine, pH of the urine falls to some extent. So pH of the normal urine is 6. This is called as acidification of urine. But in certain situations like metabolic acidosis, the pH may ranges from decreases up to 4.5, and in metabolic alkalosis it may increases up to 9.8. But normal pH is 6. Now we have seen two aims of the kidney, and these two aims of the kidney is achieved by following 4 mechanisms. First mechanism is excretion of H plus ions and regeneration of bicarbonate ions. Second mechanism is neutralization of H plus ions with the help of phosphate buffer system. Third mechanism is again neutralization of H plus ion with the help of ammonia. And last mechanism is reabsorption of bicarbonate ions. So just we have studied two aims of the kidney. These aims are excretion of H plus ion without much change in pH of urine, and regeneration and reabsorption of bicarbonate ions. To achieve these two aims, these 4 mechanisms are taking place in the kidney. So we will study in detail about these 4 mechanisms one by one. So first mechanism is excretion of H plus ion and regeneration of bicarbonate ion. The first mechanism is excretion of H plus ion and regeneration of bicarbonate ions. It occurs in proximal convoluted tubules. So what happens in this mechanism, that I will show you in the figure, then you will understand what I have written here. Now see this figure. Here I have shown 3 compartments. On the left there is plasma compartment. Next is proximal convoluted tubule, which is the cell of proximal convoluted tubule, and next is lumen of the renal tubule where urine is present. Now here what happens, we will start from PCT. In the below portion of PCT I have shown carbon dioxide combines with water to form carbonic anhydrase, to form carbonic acid in the presence of enzyme carbonic anhydrase, which get dissociate into H plus ion and bicarbonate ions. The same reaction occurs in the respiratory system, but here see the difference. In the respiratory system, the same reaction occurs, H plus ion and bicarbonate ions present in the body. But here what happens? Here H plus ions are excreted into the urine, that is into the lumen, and bicarbonate ion enter into the plasma. So this is the difference. So when bicarbonate ion enter into the lumen by crossing the cell of PCT, to maintain the electrical neutrality, the sodium ion with same charge is reabsorbed by PCT, which is filtered into the lumen through glomerulus or through the Bowman's capsule. Now this sodium and positively charged sodium and negatively charged bicarbonate then enter into the plasma again to maintain the electrical neutrality. So what happens in this mechanism? If you see, H plus ions are entering into the lumen, means entering into the urine, and bicarbonate ions are entering into the plasma, means they are entering into the body. So what is the name of this mechanism? Excretion of H plus ion and regeneration of bicarbonate ions. So here it is written, in this mechanism tubular cell absorbs CO2, combines with water to form carbonic acid in the presence of carbonic anhydrase. It breaks down into H plus ion and bicarbonate ion. H plus ions is excreted into the tubules in exchange with sodium, and bicarbonate ions are reabsorbed in the plasma along with sodium. In this way, H plus ions are excreted and bicarbonate ions are reabsorbed. Actually not reabsorbed, they are regenerated. New bicarbonate ions formed from carbon dioxide. Now what is the clinical importance of this step is, clinical importance is a drug called acetozolamide. This drug is used in various ophthalmic preparation, means it is a ophthalmic drug. It is a drug which is put in the eyes for some disease. So this acetozolamide inhibit the enzyme carbonic anhydrase. I think acetozolamide tablet is also available. So when you take this drug, it stimulate or it inhibit the enzyme carbonic anhydrase. And if this enzyme is inhibited, this mechanism in the kidney hampers. That means excretion of H plus ions and regeneration of bicarbonate ion is hampered, and therefore the patient may go in acidosis. So this you have to monitor when you prescribe this drug to the patient. Second mechanism is neutralization of H plus ions. Here the role of phosphate buffer system. Now again I will show you the figure. First you should know H plus ions which are excreted in the first mechanism, if they remain free, what will happen? They will decreases the pH of urine present in the tubular cell, and when that urine flows through our urinary tract, it may damage the urine because it is a acidic urine. So we have to neutralize this H plus ions. So to neutralize this H plus ion, phosphate buffer system plays an important role. We have already studied the phosphate buffer system and its composition. Now see this figure. In this figure, what I have shown, the first mechanism is shown below. Now what happens here is, the Na2HPO4, which is the base of the phosphate buffer system, is filtered through the glomerulus and enter into the tubular lumen. And this break into sodium cation and NaHPO4 anion. Now H plus ion, which have already entered in the first mechanism, now combines with this sodium biphosphate anion to form NaH2PO4. This NaH2PO4 is a weak acid, so it will not decreases the pH too much. So in this way, the H plus ions which are entering into the tubule is neutralized by the phosphate buffer system. The same is written here. In this mechanism, the base phosphate buffer system, that is Na2HPO4, is filtered through the filter and enter into the tubules, get dissociated into sodium cation and NaHPO4 anion. This NaHPO4 anion combines with H plus ions to form NaH2PO4, sodium acid phosphate, which is a weak acid, and does not decrease the pH of urine to a very large extent. Sodium acid phosphate is then excreted into the urine. So here instead of excreting the free H plus ion, the H plus ions are excreted by combining with NaHPO4 anion to form sodium acid phosphate. So NaH2PO4 is the major acid present in the urine. Now if you want to measure how much sodium acid phosphate is present in the urine, it is measured by titration method. For this titration, you have to collect 1 liter of urine of a patient and you have to titrate that urine with the help of N/10 NaOH until the pH 7.4 is achieved. All of us know what is titration. We have to add the N/10 NaOH drop by drop into the 1 liter of urine. So that will neutralize slowly the sodium acid phosphate and slowly the pH from 6, or if it is below 6 it will increases, and we have to maintain that titration up to the pH 7.4 is attained by the 1 liter of urine. Now after that we have to measure how much N/10 NaOH is required for this process. So depending upon that we will define the titrable acidity. So titrable acidity is defined as number of ml of N/10 NaOH required to titrate 1 liter of urine to a pH 7.4. So that ml, if we require more ml of N/10 NaOH, that means more acids are present in that urine. So in acidosis, titrable acidity is increased. The maximum limit of acidification is of pH 4.5. That I have already told you. The maximum pH that can fall is up to 4.5. Otherwise, the normal pH of urine is 6. Now the next mechanism or third mechanism is also for neutralization of H plus ions, which are excreted in the first mechanism. Here neutralization is not by phosphate buffer system, but it is by ammonia. So that mechanism occurs in distal convoluted tubule. Here trapping of H plus ion, that process is called as trapping of H plus ions. Now what will happen in this process, that I will show you in the figure. So again, this is the figure. If you see this, the first mechanism, H plus ions are excreted into the lumen in the first mechanism. Now what extra thing is shown in this slide is, in PCT the enzyme glutaminase is present. Now this enzyme glutaminase act on glutamine. Glutamine is a amino acid, all of us know. GLN represents here glutamine. So this act on glutamine, removes the ammonia from that glutamine and convert that glutamine into glutamic acid, which is also another amino acid. Now this ammonia then diffuses into the lumen, and in the lumen already H plus ions are excreted in the first mechanism. So that H plus ion combines with ammonia to form NH4 plus or ions or ammonium ions. So here the positive charge is not neutralized, but H plus ions are also not free. Therefore this is called as trapping of H plus ions. Here H plus ions are not free, but they are trapped and their charge is not neutralized. So same thing is written here. The enzyme glutaminase acts on glutamine and convert it into glutamic acid and ammonia. Ammonia diffuses into the lumen and combines with H plus ion to form NH4 ion, which is a weak acid. In this way H plus ions are trapped. Normally this glutaminase in normal condition is not very active, because in normal condition the whatever H plus ions are excreted into the lumen, they are neutralized by phosphate buffer system. But In case of acidosis, metabolic acidosis, when more H plus ions are excreted by the kidney and phosphate buffer is unable to handle this extra H plus ion load, at that time the enzyme glutaminase shows enhanced activity. That means this third mechanism is activated at this time, and whatever I have told you that acts on glutamine converted into glutamic acid and ammonia, that ammonia diffuses into the lumina and combines with the H plus ions to form NH4 plus ion or ammonium ion. So this takes 3 to 4 days to set under the condition of acidosis. But once this glutaminase get activated and this mechanism is set, it will eliminate large amount of acid from the body. We have studied how much acid is excreted in the second mechanism, or how much acid is neutralized by the bicarbonate buffer, by phosphate buffer system by measuring the titrable acidity. If you want to measure how much ammonium ions are excreted, then it is measured by addition of formaldehyde to the urine, right? Here in the in this case we have measured how much H plus ions are neutralized by the phosphate buffer system by using N by 10 NaOH by titration method. And if you want to measure how much ammonium ions are present in the urine, or how much H plus ions are neutralized by the ammonia, then you have to add formaldehyde. So total acids excreted from the body through urine is equal to titrable acidity plus ammonium ion content, and this ammonium ion content is measured with the help of formaldehyde. The last mechanism is reabsorption of bicarbonate ions. Bicarbonate is the main alkali reserve of the body. Why this bicarbonate is important? I have told you, numerous acids are formed in the body. To neutralize these acids, bicarbonate plays an important role. So it should not be lost in urine. See this mechanism, what happens in this mechanism is NaHCO3 in the lumen is filtered, that is sodium bicarbonate is filtered through the glomerulus and enter into the lumen. It is then broken down into sodium ions and bicarbonate ions. Now the H plus ions which are excreted in the first mechanism combines with this bicarbonate to form H2CO3. This H2CO3 is broken down into carbon dioxide and water. This carbon dioxide then enter into the PCT. In the PCT, the reverse mechanism takes place, that is carbon dioxide combines with water to form carbonic acid. Carbonic acid then broken down into bicarbonate and H plus ion. This H plus ion again enter into the lumen and bicarbonate enter into the plasma. So this H plus ion which have entered into the lumen is recirculated. At the same time, the sodium bicarbonate which is excreted into the lumen is absorbed in this manner, in this circulation. So this mechanism is called as reabsorption of bicarbonate ions. So this is written here, the bicarbonate ions which are filtered through the glomerulus should be reabsorbed. In this mechanism, the bicarbonate ion which is filtered through the glomerulus combines with H plus ion to form H2CO3 in the tubule with the help of enzyme carbonic anhydrase. H2CO3 or by carbonic acid get dissociated into carbon dioxide and water. Carbon dioxide enter into the tubular cell and combines with the water to form again H2CO3 in the presence of enzyme carbonic anhydrase. Inside the cell, carbonic anhydrase is broken down into H plus ion and bicarbonate ions. So here bicarbonate minus is there. So these bicarbonate ions are reabsorbed into the plasma and H plus ions are expelled into the renewable tubules for recirculation. So you remember this figure, you can explain anything. So in this process there is no net excretion of H plus ion or generation of new bicarbonate ion. The net effect of this process is reabsorption of filtered bicarbonate, which is mediated through sodium hydrogen exchanger. Sodium hydrogen exchanger means here the sodium is entering into the PCT from lumen and hydrogen ions are entering from PCT to lumen. That is called as sodium hydrogen exchanger. But this mechanism prevents the loss of bicarbonate through the urine. So in this way the pH of the body is maintained by three main system: buffer system, respiratory system and renal system. Thank you very much.